Hi, Hires. This is the first proper lesson. Um, Today, you're going to watch a sort of presentation on the structure and organisation of DNA, the first subtopic of the first topic of higher biology. Um, just before I start, if you've not got a jotter to write in, or you'd rather have you've got plenty of paper, but you'd rather have a jotter, go to the school, right? You should have had a group call by now to say that you can go and pick up jotters at the school. I think there's other locations around Broxburn that you can pick them up as well, right? So definitely do that. I think it's easier to have it all in one place. This video is going to be something for you to refer back to. Okay, so we're going to cover quite a lot of stuff. It's probably going to make your brain hurt a little bit. And then obviously you can keep revisiting this video as much as you like and pause it and rewind it and do whatever you want to do. Okay. The tasks that I will ask you to do will be in Teams and on Show My Homework like I'll always do. So when you go into Teams, obviously this is our channel, that's our chat. If we go to Files, so far there's only one thing in tasks, and that is that thing you've already done, right? The thing you did last week. There will also be pupil active notes. So basically, when I think we've done enough work and we're kind of confident, quite comfortable with the unit, I'll then get you to fill in your own notes. Okay. Now you can either print them out and fill them in by hand, or they're a Word document that you'll be able to edit, so you can do it that way if you like. And then once we've done that, there'll be past papers in there that will be arranged by topic. So you'll be able to know, I can do this little bunch of past papers now. Okay. So we'll be watching the video, then you'll be doing some tasks, then you'll be filling in the pupil notes, and then you'll be doing past papers. And we'll do that for every subtopic. Okay. I think I've told you everything about that. Okay, so let's go to the lesson and let's just get rid of me. If I can work out how to do that. Get rid of me. There we go. Right. Okay. So first thing I want to say is I want you to work hard in higher biology. You'll need to work hard. It is impossible to get a good grade if you don't work hard. But I want you to work hard for yourself. Okay, so that's what that little picture's saying. Okay, so yes, it's hard work, but you've got to want to do it for yourself. Okay, and you need to start the work now. Don't think, oh, I'll revise really well before the prelim, or just before the end of unit test, or just before we've got a check test or whatever. You need to just keep on top of it. It's not difficult stuff, but there's loads of it, so you really need to keep on top of it. Right, so this subtopic is all about the structure of DNA and how it's organised in different cells. Okay, so you'll be able to describe the structure of DNA, state the importance of the base sequence of DNA, and then there are two new terms, prokaryotic and eukaryotic. So we're going to find out what those mean and what has that got to do with DNA. Okay. Right, so you'll have seen a diagram like this before. This is just going back to basics. So we've got animals, we've got plants. Both are made up of cells. There's a very dodgy looking plant cell and an animal cell. If we look closely at a component, an organelle, a part of these cells, then we can try and figure out what the rest of these labels are. So I'm just going to give you a wee minute just to have a look at the diagram and see, do you think you know what is underneath these yellow rectangles? So just have a think. Pause the video if you like. Okay, right, so this circle here that both cells have, hopefully you've figured out, must be the nucleus. And within the nucleus, you should know from that 5 that we have lots of chromosomes. So these little red things are chromosomes, and this is one of the chromosomes. And hopefully you know that on a chromosome, you've got lots of genes. And if we look really closely at the chromosome, so we could be looking within one gene, we see that it is made of this funny looking thing, which is DNA. Okay, now you can see here that it gets its name, DNA. That's just a short version of saying deoxyribonucleic acid, right? But you are fine to always call it DNA. However, some questions in the exam might call it deoxyribonucleic acid, okay? And it is described as a double helix. We'll come to that in due course. Okay, so here I've got lots of pictures of DNA, right? You can Google it, there's hundreds and hundreds. That's a very complicated looking one, that's obviously some fancy animation that's been made, but they all show the same thing. No matter what colours they're using or what the drawings look like, they are all a double helix. Double, obviously meaning made of two parts, and helix like a spiral, okay? So you could imagine, and if we were at school, I would be getting out my DNA model and showing you this, but you could imagine that you could, if you held either side of this, you could untwist it and it would end up just looking like a ladder, right? So you can untwist the helix. You can make it just straight rather than all twisted up. But it's still going to be made of two parts, right? So if I just get my wee highlighter, we're talking about that would be one of the parts. Oh my goodness, this is really hard on the board. And it's probably going to make this a straight line. Oh no. And then the other part would be, oh, and it's continuing on going, would be that side. So that's where we get this double tear from, okay? Made of two strands and twisted up, okay? Now, if we look really closely at the DNA double helix, we see that it's made of components, right? Repeating units, they're sometimes called. And these are called nucleotides, right? So here we have, this is where it gets really dodgy. Here we have, that's me circled really badly, one nucleotide, okay? So there's one and another and another and another and another and another and another and another. Right, this is a different version. Try to show you lots of different diagrams because we don't know what type of diagram they're going to use in your final exam. So the more you're exposed to lots of different versions, you're more ready for it. You're used to sort of trying to work out what different things are on different styles of diagram. So here we can see it's all twisted up, the double helix. And at this point, it looks like it's kind of untwisted a bit so we can see what's going on. And we've got something that's got an A on it there. Let's not worry too much about that just now. This pentagon shape here and then a little red circle, okay? Those three components together make our nucleotide. Okay, so if we look at one nucleotide, so if we just go back, right? So there we've got one nucleotide and another and another and another. We're just looking at this bit here in detail because we need to know what all the parts are called. So we've got this pentagon, okay, which is our deoxyribose sugar. Now you can see where deoxyribonucleic acid gets its name from. So deoxyribose sugar, and it's called deoxyribonucleic acid. I've just taken the SE off the end there. Coming off the side of the sugar, we've got a phosphate, usually drawn as a circle, and then coming off the other side of the sugar, we've got a base. Now, you already know a little bit about bases from National 5, but you didn't need to know all the specific names of the part. So remember, this is one nucleotide. Okay, now, the phosphate is always just a phosphate, the deoxyribose sugar is always a deoxyribose sugar, but the base could be one of 4 different types. Now, I know you know these, so I'll just give you a little few seconds, a little moment to have a think. What are they called again? Now, the order that they are on the screen is irrelevant. You just need to know that we've got adenine, thymine, guanine, and cytosine. Okay, so you must know those names. You must know these are the names of the bases. You must know how to spell them. Okay, now if we go back to look at a molecule of DNA, so we have to imagine that this has been a double helix, but it's been untwisted, right? Because when it's all twisted up, it's really hard to see what we're looking at. So we've still got our phosphate, we've got our deoxyribose sugar, and we've got a base. We've got a phosphate, a deoxyribose sugar, and a base. A phosphate, a deoxyribose sugar, and a base. And you can see that they're all connected in this particular way. So between the bases, right, what is it, what is holding this nucleotide to everything else? What's holding this nucleotide to everything else? So between one base and another base, so the base of one strand and the base of the opposite strand, there is a dotted line shown here. Now that dotted line on every single one of these rectangles bases is a hydrogen bond. So that's a hydrogen bond, that's a hydrogen bond, that's a hydrogen bond, that's a hydrogen bond. And it's hydrogen bonds that are holding the base of one nucleotide to the base of the opposite strand. We've then got these brown lines. I don't know if you can see that they're brown so clear. So there's one there, one there, one there, one there, one there. They are just representing strong bonds. That's all you need to call them, okay, strong bonds. And because a bond will always form between the sugar and the phosphate, you end up with this single strand of nucleotides at this side and a single strand of nucleotides at this side, but they're joined together with these hydrogen bonds, okay? Another diagram. Again, this is similar to the one we looked at before, but this time it's got more detail about the base pairs. So here it's telling you what the colours represent. It's got the full names of the base pairs. And then on the diagram, there's obviously no space for the full word, so they've just put the first letter. And often on a diagram, it will just say T, A, C, and G. But you still must know the full names. So this one here, we've got that's our base. It calls it a nitrogen base sometimes. Don't worry about that. You just need to know it's a base. This nice turquoise deoxyribose sugar, and the red circle is the phosphate. Okay, now it doesn't matter. You could have picked that nucleotide or that nucleotide or that nucleotide. They're all made of the same stuff. Okay, so this is just to make sure we've got all the information. So we know that the four bases are guanine, cytosine, adenine, and thymine. We now know that the molecules head together in different ways. We've got the strong bonds, they were the brown bonds, between the sugar and the phosphate. And we've got these weak hydrogen bonds, the dotted line between the bases. Okay, now, for those of you that are awake, you will have noticed that the strands aren't identical. If we imagine this looks a little bit like a house with slightly bulging walls and the roof at the top there, this sugar looks like it's upside down because it is upside down. Okay, so this strand will always get in the habit of drawing it with the house the right way up, and then the phosphate at the top left, and then we've got the base here. This one is completely upside down. Okay, this nucleotide is completely upside down. So I should have really thought about this before, but what I'm going to do is go back and take this, if I can. Bear with me. So if I take that and group it and go way over to where we were. Oh, nonsense. Sorry, folks. This is just like being in a real lesson with me. Copy. And we go here. And we paste it. Right, so there's one of our nucleotides. But if we really just flip it on its head, okay, we can see that we've now made nucleotide on this side, right? So it really is just upside down. I actually turn my page upside down and draw them like I was drawing a normal one because it's just easier, okay? So the opposite strand, so this strand is not identical to this strand. How we describe it as being complementary, okay? Now notice it's complementary with an E, not complementary with an I. My spelling isn't very good, but it's not that bad. It's not playing a compliment like, oh, you look nice in that outfit. Complementary as in it sort of matches, okay? So I'm just going to get rid of that thing that I've just done. So we can see that our two strands aren't identical, they are complementary. And it's got another term that refers to it, which is being an anti-parallel structure. Anti-parallel because although they look like parallel lines, one is running in the complete opposite direction, so they're sometimes referred to as anti-parallel. Okay, right, we're going to watch a wee clip to give you a break from my voice. Okay, so that video also spoke about something that you know about from Nat 5, which is the base pair rule. So if we've got a guanine, we'll have a cytosine that pairs up with that. And if we've got a thymine, we'll always have an adenine that pairs up with that. Now, I always remember it as A and T are quite, are jagulators, as in they're made of straight lines, whereas C and G are roundulators, or curvy lines. So the curvy ones go together and the straight line ones go together. Okay, I don't think you really need that reminder because you did do it in National 5. Okay. Right, now this is where it can sort of get a little bit complicated, but we don't really need to know all the ins and outs of it. So there's our nucleotide the way we normally draw it, right, with our Houston or the right way up. Now, chemists like to give molecules numbers, okay? So if we looked really closely at this sugar, this deoxyribose sugar, we would see that it's actually made of carbons and hydrogens and oxygens, okay? And that's a carbon at that point there, that's a carbon there, carbon, carbon, and a carbon up here. And they're given numbers. So there's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Now, both of these diagrams are just taking out all the complicated chemistry bit of that and just put in a number. So there's the first carbon, the second, the third, the fourth, the fifth. This one, instead of putting the C for carbon, there's like a wee dash at the top. Now, we would say that as 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, okay? So, bearing that in mind, okay, so when we look at our housey one, this is the 3, and this would be the 5 prime end, where it's joined onto the phosphate there. If we then look at a molecule of DNA then, okay, so remember we've got our dotted lines, which are representing the hydrogen bonds, joining those two together. I'm doing this really badly. Then we know that that's 5, and that's a 3, that's a 5, that's a 3. And obviously we've already worked out that this side is completely upside down. So that's 3, that's 5, that's 3, that's 5. And if we draw our arrows going from the 3 to the 5 direction, that arrow's pointing down, 3 to 5, that arrow's pointing up. Okay, so that's just another way of explaining why are they called anti-parallel. If we number the carbons, we can see that they're going in opposite directions. Now, this 3 and 5 bit becomes really important when we do our next topic, right? But for now, I would like you just to get in the habit, just on a scrap bit of paper, maybe just drawing out that. So 4 nucleotides worth. The house on the left-hand side is always the right way up, 5's at the top, 3's at the bottom, and opposite on that side. Guys. Right, so here we've got 2 different molecules of DNA, okay? They look very, very similar, but they're not. Okay, so have a look and spot the difference. Okay, so we can pretend that this molecule has come from me and this molecule has come from Mr. Hill, for example. Okay, very, very similar, but they're not the same. So hopefully you have spotted that this bit is different for the molecules. Okay, so what is different is these 2 bases. Okay, because that's the only thing that will change. The phosphate will stay the same, the sugar will stay the same. So these bases are different from these bases. Now, on this diagram, we don't know what's A, what's T, what's C, and what's G, but that doesn't matter. I just want you to realise that the only thing that will change from one molecule of DNA to another will be the order of these bases. Oops, sorry, we've Zoomed past one. Yep. So the order of those bases is sometimes called the base sequence. So this one is going purple, pink, pink, green, green, green. Okay, that's a sequence. Whereas that was this one here, purple, pink, pink, green, green, green. This one is purple, pink, pink, green, red, green. Now remember, those colours represent the A's, the T's, the C's and the G's. So that sequence, okay, is what forms the genetic code. Okay, so what actually is the genetic code? It's the sequence of bases, and it's different for every organism. Okay, so if we looked at the base sequence or the genetic code of a human, it would be very different to that of an oak tree or an ant or an elephant or a daffodil. It would also be different between humans, right? So mine wouldn't be the same as Mr. Hill's, which shouldn't be the same as Mrs. Combs, which shouldn't be the same as yours. Okay. Right, now a really common question that you're often asked is to work out how many are there of a particular base in a molecule of DNA. Now all you need to remember is however many A's there are, there'll be the same number of T's. And however many G's there are, there'll be the same number of C's. Okay, so in this example, there are 2 T's, and therefore there must be 2 A's because they pair up. And there are 1, 2, 3, 4 G's, so there must be 4 C's as well. 1, 2, 3, 4, that's it. Okay, so here's a question for you then. In a DNA molecule made up of 200 nucleotides, so you've got 200 nucleotides in total, 40 are adenine. You've got to work out how many are there then of all the other bases. Okay, so I'll just give you a little moment, but you could always pause it if you need longer. Okay, and then we'll see if you're right. Okay, so if there's 40 adenine, there must be 40 thymine because they've got to be the same. Now that adds up to 80, which if you've got 200 nucleotides in total, that means there's 120 left, and they've got to be split evenly between the G's and the C's. Okay, so bear that in mind when we come to try some questions on this topic. Right, so let's think about where is DNA found in a cell? Now you're probably all going to go, hopefully, shout it out at the computer screen if you like. Oops, 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 why is it not coming up? You're all going to shout nucleus, I would hope, I would think. But then if you think about it from National 5, is that the only place that you find DNA? Hopefully you are thinking, hang on a minute, wait a minute, we know that bacterial cells don't have a nucleus. So for those cells, for bacterial cells, their DNA is just free in the cytoplasm. Right? It's not in a nucleus because the nucleus does not exist. Okay, so this is where we get into another little bit about DNA, how it's organised. So this slide you should mostly know from National 5. Okay, the only new bit is this word prokaryote. So a prokaryote or a prokaryotic cell is a cell that doesn't have a nucleus. Okay, now it says here prokaryotic cells do not contain nuclei. That's because we're talking about plural there. We're talking about lots of cells. So we wouldn't say nucleuses, we would say nuclei. Do not worry about that. Okay, so cells that don't contain nuclei, or a cell that doesn't contain a nucleus, you already know from Nat 5, is a bacterial cell, okay, or bacteria. So here we've got a diagram then of a very simple bacteria. Now you already know this from National 5. Bacteria have a circular chromosome, okay, one chromosome that doesn't have a start and an end, it's just one big long loop of chromosome, and it's not in a nucleus because it doesn't have a nucleus, it's just free in the cytoplasm. But in addition to that, bacteria have got these rings of DNA, these kind of bonus DNA, extra DNA that we call plasmids. Okay, so apart from this word prokaryote or prokaryotic cell, none of that's new. But we've also got eukaryotic cells or eukaryotes. And these are the cells that we learned about first in first year. Okay, these are the cells that do have a nucleus. And it says here with a distinct membrane, just forget that, just they have a nucleus. Okay, so you're thinking to yourself, well, what kind of cells are these then? Well, they're not bacteria because we've just said bacteria are prokaryotic cells. So they're pretty much everything else. So if you think about the cells you learned about in National 5, you've got your plant, your animal, and your fungi or your fungal cells. Okay. Now, some of this is new, right? Some of the next bit is new. So we know that in eukaryotic cells, like plant, animal, and fungal cells, we've got a nucleus, and that's where we find the DNA. We also know that it's chromosomes. Right? We know that we find the DNA in chromosomes in the nucleus. This word is new, linear. Okay. Now, linear means arranged along a straight line. So there is a start and an end. So we normally just draw chromosomes. You really don't have to be very good at drawing for this. There we go. There's a chromosome. It's got a start. There's a linear chromosome. It's got a start and it's got an end. Okay. So eukaryotic cells are plant cells, animal cells, fungal cells. They have a nucleus, and that's where you find their DNA within chromosomes. And these chromosomes are linear. They have a start and an end. They're just a line. Okay, so you can see where the word line comes from. There's an awful lot of chromosome to squish into a nucleus. So if we look at this picture, and there's an awful lot of DNA to squish into a chromosome. So this diagram here's got a cell with a nucleus, and then these tiny little things you can barely see are chromosomes. And if we kind of pulled the chromosome out, we would see that it would eventually form the DNA. Okay, now I don't know if you can see, but on this diagram there's also a little there's things here called histones. So imagine it's a bit like if this was a thread, right, and you had to try and ram as much thread as possible into a small space. What you might do is start spinning it around something, right, like a spool. Okay, so you would start winding the thread around something to kind of hold it all tightly in place. Now, these things that it's wound tightly around are called histones. Okay, so can you see all these little things here make up? These are called histones. And hopefully you can see here, it's like wound round these. That's all you need to know. Don't worry about, why is there, how many are there and how are they organised and whatnot. They're just DNA in eukaryotic cells is wound around histones. Okay. Unfortunately, there's a little bit more to know about eukaryotic cells. Okay, so we know that eukaryotic cells have got mitochondria and chloroplasts in them. Obviously, only plant cells will have chloroplasts, but all the cells, so the animal, the plant, and the fungal will have mitochondria. And you actually find DNA inside mitochondria and chloroplasts. Okay, so DNA can also be found in the mitochondria and chloroplasts. And we describe that DNA as circular chromosomes. Okay, now do not, this is what this wee bit here is about, don't confuse that with a plasmid. So in a eukaryotic cell, you've got the linear chromosomes in the nucleus, but you've also got circular chromosomes inside mitochondria and chloroplasts. Okay, so that's a bit weird to think about that. Okay, now there is a reason, but I'm not going to go into the reason just now. So I'm just going to scoot really quickly past the next slide. There we go. Okay, and then yeast, just to be annoyingly complicated, okay, they are eukaryotic. So they've got linear chromosomes in their nucleus, and they've got mitochondria, which have got circular chromosomes inside them. But they also, some of them, also contain plasmids. Okay, so they're just being greedy. They've got everything. They've got linear chromosomes in their nucleus, they've got circular chromosomes in their mitochondria, and in their cytoplasm, they've also, some of them, not all of them, will have plasmids. And just as a little bit of, can you remember this from Nat 5? What process then could a yeast cell like that be used for? Because it's got a plasmid, there's a process that you know that plasmids are useful for. So have a think, have a pause. I'm about to click the button here. It is genetic engineering, if your brain can remember that far back. Okay, so here we've got a kind of summary diagram, okay? So if this was DNA in a eukaryotic cell, I want you just to have a look and think what would these, what labels would be under these blue boxes. Okay, That's enough time to figure it out. So let's do this thing here. So what's circles here in the pink? That is one nucleotide. These dotted lines that are going between these two rectangles. Now, I know there's 3 dotted lines in that picture, and there was one on mine. Don't worry about that. Okay, you don't need to know that, but they are hydrogen bonds. Hopefully you were right about that. This yellow, this sort of circle here that's highlighted is a phosphate. The pentagon is a deoxyribose sugar, and this rectangle is a base. And this circular thing here that the DNA is wound around is a histone. Now, I put in brackets here protein because histones are proteins, and often the question will say, what do you call the protein that the DNA is wound around? Okay. Right. That is all the information for this lesson. Now, the first thing I would like you to do is just complete this. So just do it on a Word document or do it on a bit of paper. I want you to write down 3 keywords that you've got from this lesson. There are lots. Perhaps you could do the ones that are more new to you. I want you to write out 2 things that you've learned, 2 new things that you've learned, and something that you think, oh, well, wait a minute, what does that mean? Something you want to find out. Now, it might be something that you think I've already told you, but you didn't quite get it, or it might be something just related that's just made you think about. Okay, that's the first thing I want you to do. I will then post on the file section, like I said, of Teams, I will post tasks. Okay, but I haven't put them up yet, so I can't show you them. Right, so happy learning. Bye.